Anyway, let's start. I'm Sung-Joon Park from the Human Genome Center, University of Tokyo, and then I have the poster over here. So the please come. Sorry. We can see. Thank you. So let me make a long story too short. Anyway, in this study, I focused on the global chromatin accessibility change using the CRISPR screen technique. So as you may know, the DNA is protected from the unwanted modification by being organized the chromosome structures, right? Nucleosome structure. So for the profiling, the kind of the accessible regions, we know the ATACsee is one of the most popular technique, but it primarily focused on the nucleosome depleted regions only, which is part of the, the global accessible regions. But on the other hand, the ATACsee that utilize the fluorescent TM5 the transposon can capture the single event of the open chromatin and provide more the global view of the global chromatin accessibility changes. So in this study, we focus on how the dynamics of the global, not local, right? Global chromatin accessibility were regulated by which molecule, rather than focusing on the nucleosome depleted regions only. So we conduct some experiments using the CRISPR screening to knock out near to 2000 the human gene and assess the chromatin, assess the change in accessibility using the ATACsee signal. And then after the identifying the cells that, the dramatic change their accessibility, and then we conduct the single gene RNA sequencing for detecting which genes were knocked out in the each cell, in the cell populations. And also we perform the CRISPR, the single gene knockout using CRISPR-Cas9, the system for the comparing the pooled CRISPR the screening and the single gene knockout. We found that over 90 to 98% of genes show the consistent the ATACsee signal between the single gene knockout and pooled CRISPR screen. And also we conduct ATACseq also by the knockout of each single cell, each gene, and with some Drosophila spiking for further analysis. So here's the result. We found 102 the genes. These genes are affected in the global chromatin accessibility changes. Unexpectedly, we expect actually the enzymes like the chromatin remodeler, histone modifier will affect the chromatin accessibility, but not. One interesting finding is that the histone acetylation mediated by the CRISPR-BP or HDAC3 is very important, the feature and very important factor to regulate the global chromatin accessibility. And then, the 83 genes show the open chromatin state when each genes were knocked out, means these genes are functioning for suppressing the chromatin accessibility in wild type. So here's the category of the regulator we found. as I said, the negative regulator more than the positive effect. And also there's lots of the complexes already known to control the accessibility. And also we found the novel the molecules over there. Particularly we interested about the TFDP1 because that one is transcription factor. So we speculated the mechanics and function of this TFDP1. TFDP1 is a transcription factor, but they're forming for regulating the cells, the cellular growth by forming the complex with the others. So to make long story short, we knock out the component of the complex that we found that TFDP1 is a core one for making the open chromatin states. But the induction of reduction of the TFDP1 is not immediately affect the chromatin accessibility. We need more than 12 hours to show their effects. That means the TFDP1 is an indirect regulator. So briefly explaining, the TFDP1 knockout can increase the accessibility, but reduce the nucleosome occupancy by reducing the expression level of the canonical histone coding genes. So using this, the characteristic of the TFDP1, we transiently knock down of this TFDP1, and then apply some several biological application like the genome editing by monitoring the binding event of the Cas9, and also the reprogramming of the iPS cell by monitoring the binding of the pluripotent TFs. So actually, we found that the enhanced efficiency of the genome editing and reprogramming, iPS reprogramming the efficiency by transiently reduce this molecule. So this is summary. So we are very interested about the global chromatin accessibility changing rather than the local one. And then we found that lots of almost 100 molecules, but we typically interest about the TFDP1, and then we apply the TFDP1 characteristic to the biological application. This is a typical example how the understanding of the global chromatin accessibility can enhance the biological experiment, biological applications. So the experiments were conducted by Dr. Satoko Ishii, and then he received the PhD degree successfully, and then we got lots of the effort from other collaborators. So thank you very much. I have to take a question.
My last time to hear about ATACsee-ATACsee. What is it? Can you explain it again?
ATACsee is like a very similar, the systematically very similar to ATACseq. But ATACsee show the image fully represent the image. So ATACsee attack open chromatin region by showing light. So we can measure how the open chromatin in the single cell level.
Okay. Thank you very much for that. Any other question?
Okay, so it's a bit of a talk. Would you think the ATACsee has any potential to identify endogenous activity?
Endogenous what?
Endogenous activity.
Endogenous
the real activity. No amplification, no it's a real endogenous activity.
Endogenous. The differences between ATACsee and ATACseq. ATACsee much attack more the very narrow open chromatin regions,
okay,
like including the linker histone. But ATACseq never ever they attack actually the linker position, and also they need some narrow space rather less than 1K BP. Right? So ATACsee much, as I said, provide more the global view of the chromatin states. And then important thing is that ATACsee can measure the open chromatin state in a single cell level. Just one cell, and we can measure the fluorescent image. Right? So that one is the more the powerful thing compared to the ATACsee.
Thank you.